so thankful that Dr. Tomkins was able to come and speak to us this week. He's been here for a couple days now, and it's been a real blessing. So I hope that you guys will pay attention as he talks to us about his research that he's done. So let's welcome Dr. Tomkins.
All right, thank you so much. Let's just open up in a word of prayer real quick. Father God, we just thank you for this time tonight. I pray that you just give me your wisdom and your abilities to communicate these truths about your creation and how science supports the scriptures, and that you would just give me your grace, wisdom, and strength to do that. And I pray your blessings upon all these students, Lord. I pray that you would give them wisdom and revelation too as I'm speaking, and that you would just bless this time. And we just pray it in the mighty name of the Lord Jesus. Amen. So, let me just give you a little bit of background on myself. Someone asked me if I've ever been to Greenville before. Actually, I lived here for about 19 years, not in Greenville, but in and around Clemson University. So, let me just share my testimony real quick. It won't take too long, but I grew up in the Pacific Northwest in the Vancouver-Portland area, and I did not have a Christian upbringing. My father was a research chemist and obviously an evolutionist, and he was a huge fan of Carl Sagan. So how many of you might remember him? That was a while ago, but he was an evolutionist and an astronomer. So I remember seeing his books on the coffee table. And anyway, so that's what I kind of grew up with, but I didn't really care much about science in high school. I just wanted to party and have a good time. So I was kind of a wild character in high school, and went to a community college for a few years after that, and just kind of hit rock bottom, because you know a lot of us have to hit rock bottom before we come to Christ. And so I'm driving down the road in my '68 Mustang fastback, and I've just had enough of my life and getting in trouble and being a knucklehead. And said, God, if you are out there, please do something. And sure enough, about 2 months later, I went to Washington State University to do my final couple of years of a bachelor's degree. And I had a Christian roommate. He said, you want to do a Bible study? I said, yeah, that sounds good. I didn't know what I was getting into, but anyways, he said, let's look at some scriptures to see what happens when you die. And so obviously it was not good. I was not going to a good place. I realized I was a lost sinner, and I needed Jesus pretty bad. So I prayed and accepted Christ into my life. But anyways, the journey just started there, because right after that, I'm getting blasted with evolution from professors, from students, and so I needed some help with that. So I go to the Christian bookstore and I said, what do you have to help me with this evolution issue? And they gave me a book called The Fingerprint of God by Hugh Ross. And so I'm reading this book, and he's trying to add billions of years of deep evolutionary time to the scriptures, claiming that the creation week lines up with the geological column, which it doesn't. So I'm reading through this book, and I'm like, this does not work. This does not work at all. Anyways, I need something better than this. So I go back to the Christian bookstore, and they give me Scientific Creationism by Henry Morris II. And so I see some people nodding their heads, because that was a very impactful book, and it's still a great book. In fact, I think we still sell it at ICR. And so I read that book, and it showed how science supported the scriptures. It talked about the global flood and the fossil record and all these other things like the various laws of physics and things, and how all this supported the scriptures. And I thought, this is really awesome. So I bought everything Henry Morris literally wrote after that, and of course Dwayne Gish as well. In fact, Dwayne Gish actually came to our campus shortly after I got saved, and he debated an evolutionist and literally wiped the floor with this guy. And in fact, when he finished his talk and said, hey, are there any questions out there to the students? And there's probably over 1,000 students in there. And they all lined up, not all the students, but this massive line went up behind the microphone. Every question was for Dwayne Gish. They didn't care anything about what this evolutionist had to say. So anyway, so I had thankfully, I had a really good foundation there after I got saved to help me with my faith. But little did I know, here I would be 20 or 30 plus years later working for the very ministry that really helped me out there at the beginning of my Christian walk. So anyways, I want to talk about the monkey business in the chimp genome tonight. And this is actually an area of research I've been working on for probably about 15 years. So when I first got to ICR, well, let me go back a little bit. So then I went on to get a master's degree in plant physiology, and then went on to get a PhD in genetics at Clemson University, did a postdoc at Clemson in genomics, and then became a professor at Clemson. Actually, Virginia Tech, after I finished a couple of years of a postdoc, offered me a job as a professor, and then Clemson counter-offered, so I stayed at Clemson. And so I actually lived there for about 19 years total, in and around Clemson. My parents lived in Greenville, so I'm pretty familiar with this area. The only thing that's changed, it looks like the traffic's gotten worse since I was last here. But anyways, so I love this area. I love the upstate, the South Carolina. It's just a beautiful area. So anyways, let's get going. So Charles Darwin kind of got this whole idea of evolution going with this book on the origin of species. And of course, the title is much longer than that. People don't realize that he wrote another book called On the Descent of Man, where he claimed that humans evolved from an ape-like creature. And so that was very impactful on the scientific community during that era in the late 1800s. And these three guys helped to kind of push that idea even more. So Ernst Haeckel was a professor in Germany, a scientist in Germany, and he kind of had his own ideas about evolution, but he still believed that humans evolved from an ape. And then, of course, Charles Darwin was pushing his ideas on human evolution as well. And then Thomas Huxley was known as Darwin's bulldog, so to speak. He was his PR guy, as it were. And so he promoted these evolutionary ideas, including human evolution, to the general public. And so that's how things really got going there in the 1800s. And eventually that evolved, so to speak, evolved into what we know as neo-Darwinism today, where evolutionists believe that through random mutations, they somehow magically create wonderful new traits, and then these new traits are magically selected for by the environment, and that's how evolution proceeds. But as I was talking in another talk I gave a couple nights ago, that idea now is coming along under a lot of scrutiny, not by creationists, but by evolutionists themselves, but that's another talk. So where do evolutionists think modern humans came from? So how many of you have heard of the out of Africa hypothesis? Okay, so depending on which evolutionist you're talking to, they think modern humans, anatomically modern humans, came out of Africa 100,000 to 200,000 years ago. But there's some major problems actually with this idea, and that happens to be this scientific data. In fact, they have found human fossils around the world that completely contradict these dates. And so Homo erectus fossils, which are actually human fossils, now evolutionists call them archaic humans, meaning just humans, old humans, have been found to completely contradict out of Africa idea. So they have found Homo erectus fossils in China that they supposedly dated at 800,000 years ago. Now, I don't accept these dates, but I'm just showing you how even the evolutionists' own data debunks evolution. If that wasn't bad enough, they supposedly have found Homo erectus fossils on islands in Southeast Asia they claim were 1.7 million years old. Well, I thought humans came out of Africa 100,000 to 200,000 years ago. So you can see the data never really matches up with evolution. It's all very speculative and hypothetical. In fact, this guy, Bruce R. Fenton, is an evolutionary science writer in Great Britain. And because of the data that I just showed you, in addition to a lot of other data, he wrote this book, The Into Africa Hypothesis. And so he says, based on all the data, it looks like humans evolved outside of Africa and then went back into Africa. So anyways, so why Africa to begin with? Well, this is the only place where you find chimpanzees. And evolutionists think that humans evolved from a chimp-like creature or a shared ancestor with chimpanzees. And so that's what got that whole thing going. And in fact, they believe that chimpanzee is the closest living ancestor to modern humans. And so if you were to see a standard, what we know as a phylogenetic tree or an evolutionary tree, I see some of you laughing, you recognize John Wayne there, so you're right at the pinnacle of evolution, right? Or we could put Clint Eastwood up there, right? Anyways, so this would be a standard evolutionary tree. So, in other words, humans are most closely related, supposedly, according to chimpanzees, and then gorillas, and then orangutans, and then there's a rhesus macaque as an outgroup there at the bottom. So that's the standard pecking order, so to speak, in human evolution that you will normally hear about. However, not all evolutionists agree with each other on this. In fact, there was a paper published, I believe that was 2009, and the title of the paper is The Evolution of the Second Orangutan Phylogeny and Biogeography of Hominid Origins. And so in this paper they said, no, humans are not most closely related to chimpanzees. They're more closely related to orangutans because we share more anatomical features with orangutans than chimpanzees. The fact of the matter is, the chimpanzees, orangutans, and gorillas and humans, they're all unique. So there isn't any distinct evolutionary relationship between any of them. I believe that the three different kinds of great apes are created kinds, as the scriptures say. In Genesis, everything was created after their kind. Of course, humans were created in the image of God. And so I actually began doing some evolutionary trees myself. I began doing it for protein sequences because proteins tend to be most similar among mammals. And so I'm thinking, well, will this give me an evolutionary tree? And it turns out, here's a protein called alpha motif protein 11. And in this particular tree, humans were most closely related to gorillas. And then macaques. They shouldn't even be related to macaques at all. But there's macaques down there right below gorillas. And then orangutans and chimps are at the bottom. And so I began doing more evolutionary trees with protein sequences. This is an ACAPS protein. And there again is gorilla at the top, rhesus macaque, and then chimps. And then at the bottom was orangutan. So the fact of the matter is it's almost impossible to get a clear evolutionary tree using protein or, as we'll see, genetic data as well. In fact, this paper was published in 2012 when the gorilla genome, or at least the first draft of the gorilla genome, was first published. And they said this in the paper: in 30% of the genome, gorilla is closer to human or chimpanzee than the latter are to each other. And then this paper, which was published in 2007, where they actually looked at evolutionary trees between the great apes and humans, they said this: for about 23% of our genome, we share no immediate genetic ancestry with our closest living relative, the chimpanzee. So the fact of the matter is it's hard to develop any good evolutionary tree with the biomolecular data that we have, because it seems like that everything is sort of a mosaic, if you will. Yes, we share some traits and similar with orangutans, some with gorillas, some with chimps, but there's no clear evolutionary pattern. So what about this whole idea, which is probably why a lot of you are actually here today, with the idea of humans and chimpanzees being 98 to 99% identical? And I don't need to go back to my story where I was talking about my testimony. So when I left Clemson in 2009, I'd been there working as a professor in genetics and biochemistry, running a genome center for about 10 years. And I just got to the point where I felt like I needed to do more with my life than just kind of laying low and doing genomics research, because you can't be open about being a creationist in creation science without getting fired at a major secular university. It's just the way it is, unfortunately. especially if you're involved in the life sciences. So anyway, so I get to ICR in 2009, and the president of ICR at the time says, we go to churches, you know, around the United States, and people are always asking us, are we 98, 99% similar to chimpanzees? And he said, I need an answer, and I want one in 6 months. And there was two biologists at the time, myself and one other guy, and I had just gotten there, so I didn't know what to say to this guy. And the other guy said, Henry, and the president's name was Henry, it's going to take more than 6 months. And then he left and went to work at a Baptist university in Louisiana. So here I am by myself, and so I'm kind of diving into this. And of course, I didn't know what I'd find. I was completely open as to what I would find in the data, which is what I'm going to share with you today. And so that began actually a journey that lasted close to 15 years. And because there were so many kind of ins and outs to this research, and so many things I ran into that were pretty interesting. Anyway, so let's get into it. So one of the first evolutionary papers where they actually attempted to look at the DNA similarity between chimps and humans was at the very beginning of the Chimpanzee Genome Project. So at this point in 2002, they had several million chimpanzee DNA sequences, and they queried those against the human genome, and this is what they said. They said about 2/3 could be unambiguously aligned to DNA sequences in humans. In other words, only 2/3 of these chimpanzee DNA sequences could they actually match up onto the human genome. This is at the very beginning of the Chimpanzee Genome Project. Well, when I began digging into it, I'm seeing papers like this, but I also want to look at the papers where they're actually claiming a 98 to 99% DNA similarity. And so I looked at the 6 top evolutionary papers that made that claim. And actually, Jerry Bergman was a co-author on this. And the main thing that Jerry contributed was he was the one that instigated me to do it. So anyways, I was actually the one that basically did the research. And so I'm looking at these 6 different papers, and the one thing that I found is that depending on the paper, if I was to factor data back in that they threw out or didn't include, I was coming up with genome similarities between 66 to 87%. And some of the papers, I couldn't even factor the data they threw out back in because the materials and methods are so sloppy and ambiguous, I couldn't even do it. And so at this point, I'm thinking, you know, it's obviously, they're just lining up sequences that are highly similar between humans and chimps. They're throwing other stuff out. You know, they're not including everything. There's something weird definitely going on here. And definitely some monkey business, as it were, going on in the chimp genome. So let's do a little history on genome sequencing concerning humans and chimps. So the human genome, the very first draft of the human genome, was published in 2001. And there's Francis Collins there shaking Craig Venter's hand. So Francis Collins was in charge of the public sector of the Human Genome Sequencing Project. He worked for the NIH, and so he headed up the public sector of it, and then Craig Venter headed up the private sector, and he was the president of Celera Genomics. And so they agreed together to basically share resources and then publish their own versions of the human genome paper in separate journals at the same time. So one of them published in Nature and the other one published in Science, which are the 2 top-tier scientific journals. And so that was done in 2001. Let me first of all say they had billions of dollars to do this research. And so they went about it very methodically and very thoroughly. And then the chimp genome got funding, but it didn't get that much funding, not nearly as much as humans, because we want to sequence the human genome with great accuracy, right? Because we're interested in solving all these human disease problems like cancer and heart disease and diabetes and things like that. And so the chimp genome really didn't get that much money, but they published the first draft of it in 2005, was when the first draft of that was published. So when the chimp genome was published in 2005, I got a hold of that paper and I began trying to look into it and say, where is this 98.5 DNA similarity? Well, it looked like it was present in some of the alignments they did, or matching chimp onto human. But it was obvious that they were not including a lot of data. And so when I factored back in the data that they tossed out, I was coming up only with 81% DNA similarity between chimps compared to humans. So how have evolutionists achieved estimates of high similarity between chimps and humans anyways? Well, they've omitted the non-similar regions of the genome. And some of this is just due to the algorithms can't match up these regions because they're so different that these algorithms can't handle that data. But on the other hand, they're also throwing out a lot of data. And these evolutionists are only comparing the regions of the human and chimp genomes that are highly similar. And like I said, some of that's just due to the constraints of the available algorithms. But, you know, there's other aspects too that they're just disregarding and throwing out data. So what do we call that? We just call that cherry picking. So in other words, these high similarity figures are just based on cherry picking the data. And you know, if any of you go on to graduate school, you'll basically read papers where it's obvious people are cherry picking the data, whatever the research is, because they want their data to look good. So anyways, let's talk a little bit about DNA sequencing. So DNA sequencing, unfortunately, is not like Star Trek, where you go to some planet, you know, and you find some weird creature and you extract a blood or a tissue sample, you stick it in the computer, and out pops a genome. So it's not like that at all. It's actually a very laborious and tedious process, especially 20 to 30 years ago when things really got kicked in concerning genome sequencing. So let's talk about the human genome sequencing project. So if you think of a genome, or the human genome, as a set of encyclopedias, maybe each encyclopedia corresponds to a chromosome. And so in the human genome sequencing project, they had billions of dollars. They had lots of labs involved in it. The NIH in Washington, D.C., this huge genome center in Seattle, one in St. Louis, one in Tokyo, in Europe. And so they had all this money, and they just literally went page by page by page, so to speak. If I was to get into the technical details, your eyes would glaze over. But anyways, that's a simplified version of how they did it, very methodical, so they could get a really clear picture of the human genome, because they had billions of dollars to do it. So we could call that a page-by-page methodical sequencing strategy. Well, the chimp genome, they didn't have that much money, and so they did what we in the business of genome sequencing call shotgun sequencing. How many of you heard that term at some point? And so in a shotgun sequencing project, or an all-at-once sequencing strategy, that would be like taking an entire set of encyclopedias and throwing them in a paper shredder and then trying to somehow put that all back together. Because during this period of time in DNA sequencing, we could only generate, with the chemistry that we had, DNA sequences that were in snippets of about 700 to 1,000 bases or nucleotide letters long. And then you have to assemble those and put them together to recreate chromosomes or genomic regions. And so with the chimp genome, they didn't have these billions of dollars to be real methodical about going about it. So how do you think they actually sorted all that out? Well, think about putting together a jigsaw puzzle. Now, I'm not a big jigsaw puzzle guy, but a lot of people are. And in fact, my wife's parents who are retired, and they love putting together jigsaw puzzles. So how do you do it? Well, you look at the picture on the box, right? I mean, otherwise it'd be really hard to put the thing together. Or there's a little poster that comes in there and you look at that. Well, with the chimpanzee genome, they had all these snippets of DNA, so how do you think they put them all together? Well, they said, let's use the human genome as a scaffold or a guide to put all these chimp sequences onto. And so that's how they did it. And they just assumed, oh, we evolved from a chimp, you know, just 6 million years ago, and you know, our genome should be really similar, like 98, 99% similar. So let's just use human to sort this mess out with. And so that's exactly what they did. And so that would be the human genome, a section of a chromosome there in blue. And then they would take the chimp DNA snippets and line them up onto that chromosome. And then they would end up something like this, where they would have chunks of chimp sequence lined up on a human chromosome with gaps in between them. And so that's how they went about doing that. Well, what's really interesting, when DNA sequencing technologies improved and the chemistries improved, where they could create really what we call long-read DNA sequences or long-read DNA sequencing, much longer than 700 to 1,000 bases long. They published a paper in 2018 where they resequenced the great ape genomes. And they actually admitted to something very important that I had been talking about for years in talks and in papers I published. And they said this: the higher quality human genome assemblies have often been used to guide the final stages of non-human genome projects, meaning the great apes, including the order and orientation of sequence contigs, and perhaps more importantly, the annotation of genes. Well, it's worse than just the annotation of genes. I actually found a web page at a genome center in the UK where they actually inserted human genes into the chimpanzee genome electronically. It didn't exist. They just thought they should be there. I'm like, you gotta be kidding me. I mean, talk about faking it. But anyways, they said this in the paper: this bias has effectively humanized other ape genome assemblies. In other words, gorilla, orangutan, chimp have been humanized, and they admitted it. Stuff I had been saying for years in talks and in papers. Another problem we have is something known as human DNA contamination. And several papers have been published, one in 2011, one in 2016, where researchers began to realize that a lot of databases, public databases, and by the way, all these DNA sequence databases, for the most part, are public. You can go to them, you can extract your own sequence because your tax dollars pay for these sequences. And I had a number of NSF grants, NIH grants, and you are literally required by the funding agency to submit your DNA sequences within just a few months to this national database at the NIH and CBI. And so researchers began to get concerned about human DNA contamination in these databases, so these papers were published, and they found out that databases from everything from bacteria to fish were full of human DNA. And a fish genome, which is a model fish genome, which is the model fish genome, they found that 3 million bases on chromosome number 3 in zebrafish was entirely human. 100% human. So how does that happen? Well, it's because human DNA is getting in these sequencing reactions. Or at least it did back in the 1990s and the first half of the 2000s. So how does that work? Well, I know what people do in the lab. I ran a DNA sequencing lab for 10 years. And, you know, people are at their bench, you know, getting their reactions together, and they're sitting there talking to each other and laughing and carrying on and coughing, sneezing. Human DNA is flying around in the atmosphere through the ventilation system. And so at that time, you know, we did not realize what a problem that was. And so anyways, so I'm beginning to wonder, well, what about the chimp genome? What's going on with that? Is there human DNA in the chimpanzee genome? So I had questions. Is the chimpanzee genome first? Is it accurately assembled? Well, no, because it's done on human. And is human DNA contamination a problem in the chimp genome? I think these are two valid questions based on all the papers I read and the problems that were popping out in the literature. And so what is the actual DNA similarity between humans and chimpanzees, and how can this be determined? Well, first of all, let's talk a little bit about DNA similarity, and then we'll get back to the issue of human DNA contamination. So here's a theoretical sequence from human and chimp. And so you have the 4 DNA letters there, nucleotides. and TCAG. And so they're the same in these two theoretical lineups here, or alignments. 100% identity there. But let's say you change one base. It's a C there in red in human and an A in chimp. And so that'd be 27 out of 28 bases match. That'd be a 96% similarity. Another question is, what gets included and what gets thrown out? So we call those single base changes substitutions. In other words, a single base has been substituted for something else, but we have more going on than that. We have these things called indels, which mean insertions and deletions. And indels can vary anywhere from just a few nucleotides to hundreds to even thousands of nucleotides. And so in this hypothetical example here, if we just look at the substitutions, we're finding just a 98% chance. And a lot of times the evolutionists would set their algorithms to avoid gaps, so as all they were reporting on was substitutions in these alignments. But you can actually set these alignment algorithms to include gaps, and so if we were to do that here in this particular example, the substitutions plus the indels, there would be 39 out of 48 nucleotide matches, or only 81%. So this is how we go about trying to ascertain and find out DNA similarity. So I actually did a study, and this was published in 2016 in the Answers Research Journal, and I looked at all 101 of the chimpanzee DNA data sets that went into the initial stages of the chimpanzee genome project before they changed over their sequencing technology. And so I was looking at DNA databases produced between 2002 and 2011. And so I sampled at random 25,000 DNA sequences for each of these 101 data sets. And I ended up with 2.5 million chimp DNA sequences that I queried against the human genome. And on average, these sequences were about 700 bases long each after I trimmed off the ends that were low quality and trimmed off what we call vector sequence, which sometimes gets in there as well. So I trimmed up the DNA sequences so they were high quality, queried them against human. And because this data also had metadata attached to each sequence, I was able to also analyze the metadata. So in these early stages of DNA sequencing, what we call Sanger-style sequencing, they would also add on the quality values of the electropherograms for the sequence. They would add on the location where the sequence was produced, and they would add on the date the sequence was produced, is what I was really interested in. So I set up a relational database to access that data like that. So when I did my querying, I would just take the very best hit, because if you take a chimp sequence and you query it against the human genome, you'll get hits all over the place. So I just took the best one. And then when I looked at the metadata, I saw something really bizarre. The DNA similarity between chimp and human started off pretty high in 2002, and then it dropped. And look where it ended up in 2011. So what in the world is going on here? So between 2002 and 2004, the DNA similarity between these sequences between chimps and humans was 92%. But the DNA sequences produced between 2005 and 2011 was 85%. That's a 7% difference. So what do you think that's due to? The contamination, right? So, in other words, all these chimp DNA sequences that were produced in the first half of the chimp genome project were contaminated with human DNA, so that they were 7% more similar to human than the ones produced in the second half. I mean, it's just like opening a can of worms when you get into this stuff. It's like, how do you get at the real numbers, you know? And so I kept hitting stuff like this, and I'm just like, you know, when am I going to get to a final answer here on this? So I did another study in 2018, and by this time the DNA sequencing technology had changed, the chemistry had changed, Now they're using this long read DNA sequencing technology. There's several different chemistries depending on the company that's selling it. And so at this point, it changed everything, because the DNA sequences we can produce now can be up to 50,000 bases long. That's a long piece of DNA. And if something's, say, 30 to 50,000 bases long, you can tell right off the bat if it belongs to chimp or human. So anyways, at this point, the human DNA contamination problem had been, at least for me anyways, had been largely solved. And so I downloaded these new high-quality long DNA sequences, and I queried them against chimpanzee. I selected 18,000 of these at random. And you can do this with computer programs that have, random number generators, so it's not that difficult. So anyways, and so the average length of each of these DNA sequences was 30,000 bases long, so they're pretty substantial. As it turned out, I could only match up 10,000 bases of those 30,000 bases onto the human genome before the algorithm hit a gap so big that it couldn't keep lining up the DNA or aligning it. So anyways, so on average, these alignments were about 10,000 bases long. Unfortunately, they weren't 30,000, but 10,000. But anyways, so the average identity of these alignments, though, these 10,000 bases out of the 30,000, was 84%. In other words, 84% DNA similarity chimp compared to human. 84%. Using this brand new DNA sequencing data that's a lot more accurate. And what's really interesting, within the same 6-month period that I published my paper on that study, Richard Buggs, who is a very famous evolutionist at the University of London, Queen Mary, did his own study, and he actually downloaded all these DNA alignments between chimp and human that were available at the UCSC genome browser, and he determined the percent identity, or the identity of chimp compared to human. And this guy's a famous guy. He's actually the editor for Nature Ecology, and a very well-respected researcher. And he also has a blog, his own personal blog, that he puts up stuff. And he also puts up stuff on the Nature Ecology blog as well. And so he said this in 2018, roughly about the same time I published my research. He said the percentage of nucleotides in the human genome that had 1-to-1 exact matches in the chimpanzee genome was 84.38%, 84%. Same number that I came up with. So I'm feeling pretty positive at this point, but I'm also thinking, what else can I do at this point? I'm getting, ridiculed by evolutionists because I'm this crazy creationist. Of course, no one ever ridiculed Richard Buggs, but I got ridiculed because I'm an open creationist. But at this point, I'm like, there's nothing more I can do. I just need to wait and see what happens out there in the secular evolutionary community. Maybe they'll catch up and admit the truth. Well, sure enough, last year, 7 years after I published that paper, in April 2025, there was a paper published in Nature, and it was the same group that did the 2018 paper, the University of Washington Genome Center up there in Seattle. And what was really interesting was the paper was 17 pages long, the paper in the journal, and they just really skirted around what the DNA similarity was genome-wide between chimps and humans. And I'm like, this is really weird. In fact, they were trying to push this evolutionary meme, and it was just, I'm like, this isn't adding up. And then they said, if you want to look at more data, you can go to the supplemental material. Well, the supplemental material was 171 pages long. I mean, it's a literal book. And so it took me a while to go through all this supplemental material. And then I began hitting these tables where they were giving figures. And and what's really crazy is that for a haploid genome equivalent from chimpanzee queried onto the human genome, they said there were 85% nucleotide identity It was an 85% nucleotide identity. But then when they queried a diploid complement of the chimpanzee genome, which is even more accurate than, say, a single genome equivalent, they said that the chimpanzee genome had an 84% one-to-one autosomal, which are the non-sex chromosomes, DNA identity compared to human. Now, the chimpanzee X chromosome, which is the female chromosome, that was the same as the autosomes, 84%. But the chimpanzee Y chromosome was less than 30% similar to the human Y chromosome. So this whole study, yeah, you can see why they buried all this stuff in the materials, or the supplemental material, because it completely trashed evolution. So why is this even important anyway? Some people might be saying, well, what's the evolutionary problem with 84%? Well, theoretical evolutionists need a 98.5 to 99% DNA similarity for their theoretical models of human evolution to work, and it's based on what we call the neutral model theory of evolution, which I won't get into, but I've talked about in some of my other talks. So for their statistical evolutionary models to work, where they can go back to the human ancestor roughly 6 to 8 million years ago, they need a 99% DNA similarity for these models to even work. And so basically an 84%, and of course it's much lower than that, because those are only the nucleotides that match, that 84%. That doesn't include the nucleotides or the regions of the genome present in chimp but absent in human, or present in human but absent in chimp. That doesn't even include those. So who knows what we get if we factor those in. I'm just guessing off the top of my head based on my own experience. The real genome-wide similarity would probably be closer to 75% or maybe even less. So, how many of you have heard of John Sanford? So he was a professor at Cornell University, and he did not become a Christian until late in life. He was an evolutionist for most of his secular career, and then it was actually creation science that brought him around to becoming a creationist because of all the data that was out there supporting the literal Genesis. But he did a lot of research in his later years after he became a creationist on what he called genetic entropy. So what does that mean? Is the human genome evolving and getting better? It's devolving and getting worse. So Adam and Eve's genome was pristine. There was no mutations in it whatsoever. But over time, mutations began creeping in, primarily because they disobeyed God, fell into sin, brought a curse on creation and on the earth. So literally every creature's genome on the face of the earth, including mankind's, has been devolving for the past 6,000 years, and probably getting much worse after the global flood, because we know that the conditions before the flood were much more favorable to life. There's higher oxygen content in the atmosphere, there's higher air pressure, there was a lot of plant life that we no longer have anymore that was probably providing critical nutrition to longevity. And so the human genome is not evolving and getting better. In fact, we're all carrying what we call a mutational load. So we're all actually, every one of us is carrying mutations in our genome. That's why by the time the Levitical law came around, God told Moses to write it down and tell the people of Israel you cannot marry close relatives anymore, because the mutational load had built up at that point to the time when it was dangerous for you to marry even a close cousin. And we know Abraham married his half-sister, but that was way before the law was given. So, a lot of this stuff that we see in the Bible has a scientific reason behind it. Yeah, Moses at the time knew nothing of genetics, but here we are several thousand years later, and we can say, wow, the Bible makes perfect sense. And so the Bible is not necessarily a scientific textbook, but the statements in the Bible are scientifically true. And so this is just one of them. So the human genome is not evolving and getting better, it's actually devolving. And so we can also see this by the lifespans of humans after the flood. So before the flood, humans were living to be about 800 to 900 years, which is really amazing. But after the flood, their lifespans just plummeted. And I think a lot of this is due to genetic entropy. Of course, I think a lot of it's also due to the environment being much more harsh and unfavorable to optimal life as well, and other things. But the human genome is definitely not evolving and getting better. In fact, there are some evolutionists that even admit this. In fact, there are some evolutionists that wonder why we're even still alive with all the mutations we have in our genome. They think that we should have been dead, hundreds of years ago. So anyways, here's a couple of papers that were published showing how mutations essentially affect the human genome, and they also support a biblical timeline. So there's kind of several benefits from these papers for creationists. But let's talk about the devolution first. So the people that did these studies were interested in looking at the rare variants in the protein coding regions of the human genome. And so these protein coding segments of protein coding genes are called exons. And so exons for their protein coding comprise about 2% of the human genome. And we can amplify these regions up and sequence them and look for mutations, which is exactly what they did. So they looked at over 1,000 people in North America in one paper, looked at over 1,000 people in the other paper in Europe and the Middle East. And they found out that over 80% of these rare variants in these protein coding regions are detrimental to human health. In other words, they're connected and associated with heart disease, with diabetes, with cancer, with things that are not helpful at all. And so that just goes to show you that mutations do not produce anything good. So the evolutionists basically have no mechanism. If mutations are doing bad things in the genome, then there's no mechanism for neo-Darwinism to even work. But the really cool thing about these papers is they looked at these rare variants, and they wanted to create molecular clocks for these variants. And so instead of calibrating their molecular clocks with deep evolutionary time, which is purely hypothetical and theoretical, they calibrated these molecular clocks with how fast populations grow, with how many children these populations have. So it was demographic data that was empirical. It wasn't a bunch of theoretical, deep time nonsense. It was real world data. And guess what they found out? That these variants, when put into a molecular clock scenario, only went back 5,000 to 10,000 years. Yeah, I literally about fell out of my chair when I read these papers. That's a biblical timeline. So the date of creation from the present back to 6,000 years completely lines up with these papers. So once again, science supports the scriptures, and we have these detailed chronologies and genealogies in the Bible. And a lot of you probably, you read through your Bible and it's like, oh, another genealogy, another chronology, how boring. Well, it's amazing how detailed they are. And they're in Genesis, they're in the books of Chronicles, and in various places, even in several of the Gospels. And so these chronologies are highly detailed, highly accurate, and they give a timeline to creation 4,000 years from Christ to the creation. So anyway, and now scientific data is completely supporting that. So let's briefly talk about human evolution, because this is directly connected to this whole scenario. And so what do we see in the fossil record? Basically, we just see apes and we see humans. There's a lot of problems with that, which is the scientific data. And so the original ape that supposedly morphed into the proto-human is known as Australopithecus. There's different species of that, supposedly. The problem is that we have human tools, artifacts, and footprints that go back well before Australopithecus creatures supposedly lived. So Australopithecus creatures supposedly were about 1 million to 4.5 million years ago, yet we have modern human-type artifacts that go back even more than that. And on top of that, we have human bones, including Australopithecus bones and modern human bones and things that also overlap. And so we don't have a timeline for really human evolution to even make sense based on these supposed fossils. So there's a number of things that debunk the human evolution story from the paleontological record. One of them is the Laetoli footprints, which were discovered in 1978 in Laetoli, Tanzania, and they dated these at 3.5 million years old. So who made these footprints? Well, human and chimp footprints are pretty distinct from one another. There's a human foot and a chimp foot right there. And you can see the chimp foot is made for climbing trees and swinging around in trees. There's the Laetoli footprint, matches up completely with the human footprint, and they dated that at the same time that these Australopithecus creatures were supposedly running around, or swinging around, I should say. In fact, there's an evolutionist that said this. He said these footprints were indistinguishable from those of habitually barefoot Homo sapiens in the Journal of Physical Anthropology. One of the most important finds, I think, is what is known as Old Dubai hominid, number one. It was an anatomically modern human that was found in 1913 in the same supposed 3 million year old strata as Australopithecus fossils. In other words, and it was almost a complete skeleton and looked exactly like a modern human. And in fact, the guy that dug it up was so concerned that people would say, oh, you just discovered a recent burial. He actually had a geologist come in and verify that this was, in fact, a human that was buried in these sediments and not ritually buried. And so since then, evolutionists have basically just swept this under the rug and they never talk about it. And then we have Lucy, and I remember actually Donald Johanson came to Washington State University while I was there with Lucy. And that was discovered in Ethiopia in 1974. The bottom half of Lucy is highly debated. It looks like they may have actually added on some human child bones to the bottom, but the top half of is completely ape-like. And this fossil is, this assemblage is only 40% complete. They dated it at 3 million years, and they claim she was bipedal, in my opinion, because they tagged on some human bones on the bottom. So did Lucy walk upright if we exclude the human bones? Well, this guy named Eric Stockstad, a famous evolutionist and paleontologist, said this: I walked over to the cabinet, pulled out Lucy, and Shazam. Isn't that what Gomer Pyle said? She had the morphology that was classic for knuckle walkers. So when chimps walk around on the ground when they're not in trees, they have a specialized wrists that lock, and then that's how they walk around, because that's how God created them. We don't have that, and it's a very distinctive anatomical feature. This other guy, I love the title of this article, Lucy on the Ground with Knuckles, Science News. And they said, A, Australopithecus anamemsis and Australopithecus afarensis, the latter represented by the famous skeleton known as Lucy, have wrists capable of locking the hands in place, during knuckle walking. And again, here's one more. Evidence that humans evolved from, oh, I want to read the title to that one. Lucy on the Ground with Knuckles. So that's a playoff of the Beatles, song back in the 1960s. Anyone thought that was funny. So here's one. Another, this was in Nature in 2000, and it says, regardless of the status of Lucy's knee joint, once again, the bottom half of her is very controversial, new evidence has come forth that Lucy has the morphology of a knuckle walker. So what about Australopithecus? What do evolutionists actually think about it? Is it really the beginning of human evolution? Look at this article, published in 2016 in the Royal Society out of UK. This is once again, a very top-tier journal. And these evolutionists said this. And I love the title of the article, From Australopithecus to Homo, the transition that wasn't. And they said this in the article, although the transition from Australopithecus to Homo is usually thought of as a momentous transformation, the fossil record bearing on the origin and earliest evolution of Homo is virtually undocumented. really about the only thing we have in between apes and humans is this character originally known as Homo habilis. It was discovered by Louis Leakey, but it turns out it was just a complete fraud. And this is from Nature in 1995, once again a top-tier journal. And they say now Homo habilis is widely considered an invalid taxon, a mixture of ape-like and human fossils. And I love this quote from 1992 from the American Journal of Physical Anthropology. If there weren't a Homo habilis, we would have to invent one. So anyways, that you can X this out as any sort of transitional form. So basically we have nothing between apes and humans. What about these Neanderthal characters? Well, you basically wouldn't know a Neanderthal unless he came up to you with a name tag and a spear. If he was to walk by you with a business suit on, he would look just pretty much like anyone else. And so this is now how they're showing Neanderthals. And besides the mean look on their face, they look just like modern humans. And so Eric Trinkus, he had an article that's interesting title, Hard Times Among the Neanderthals, published in Natural History. He said this, and he's a famous paleontologist. So he said, detailed comparison of Neanderthal skeletal remains with those of modern humans have shown there is nothing in Neanderthal anatomy that conclusively indicates locomotor, manipulative, intellectual, or linguistic abilities inferior to those of modern humans. So the Neanderthals were just humans. So here's Svante Pavo. He's become quite the rock star in the area of paleontology because he is now sequencing DNA from Neanderthal skeletons. And so how do we get DNA from a Neanderthal? Well, they're ritually buried inside caves in a dry environment, so water is not percolating down through the soil, and so the DNA is considerably more accessible and pristine than, say, something that was just buried outside in the dirt. So anyway, so they are able to extract DNA from these. It is highly degraded. I have actually worked with Neanderthal DNA myself. You can download it for free as well. And I actually had a postdoc at the Max Planck Institute that was giving me access to their genomes that weren't even published yet, which is a kind of a miracle. They knew I was a creationist. I wasn't trying to hide anything, but they gave me access, and of course it was just human. Actually, I got a funny story about the human DNA contamination thing. So I started working with this one genome they gave me access to, and I thought, well, I'll just start with the Y chromosome. It's very small. That'd be a good place to start. And I told them I'm working on the Y chromosome. I get an email from the postdoc that gave me access to these sequences a couple weeks later, and she said, "Stop looking at the Y chromosome. We think it was a girl." And it turns out that, yeah, probably human DNA contamination. Probably came from one of the lab workers, or maybe one of the guys that was working on the dig site, or who knows. Anyways, so now we see articles like this popping up with entertaining titles to them. Here's one: humans bred with this mysterious species more than once, new study shows. Well, how about that? Humans breeding with humans. And your Neanderthal DNA might actually be doing you some good. And there's actually a funny story behind this one, because Neanderthals had larger brains than modern humans. And so there were a lot of molecular biologists saying, we need to clone Neanderthal brain genes and stick them in modern humans so we can make our brains bigger and be smarter. So that kind of goes against, the evolutionary mindset, doesn't it? And then here's one: ancient humans have been mothers to some Neanderthals earlier than we thought. Well, it turns out in caves in Israel, they have found Neanderthal skeletons buried with anatomically modern human skeletons and everything in between, because they were all interbreeding and having children together. So, and in fact, they've also discovered genes for red hair in Neanderthal DNA as well. So what about Homo erectus? We're getting to the end here, so don't stress out too much. So what about Homo erectus? Well, this is just another type of what evolutionists are now calling archaic humans, meaning ancient humans. And so Homo erectus fossils are very similar, the skulls anyways, are very similar to Neanderthals. They have a sloping forehead and a large brow ridge, and we have many skulls for these. And in fact, their brain size is in the range of modern humans. Neanderthals would be bigger than that. And so they're just humans. And so here's Turkanaboy that was discovered in 1984, and there's the artist's reconstruction of him. I don't know why they made him look mean and put mud all over him, but who knows? Maybe he was mud wrestling, I don't know. But anyways, the interesting thing about Turkanaboy, he was labeled as Homo erectus because his skull had a sloping forehead and a large brow ridge, but everything below the skull, and this was a pretty intact skeleton. Almost everything was there. Everything below the skull was anatomically modern human. Totally human. So Homo erectus was just human. So Neanderthal, Homo erectus. So two of the main features on the skull are a sloping forehead and a pronounced brow ridge between Neanderthals and Homo erectus. Well, it turns out these are still present today among humans. So this is a guy who was sitting in a dental chair in South Africa. So this dentist in South Africa, who's a creationist, takes pictures of his patients that have archaic features, or allegedly archaic. So I don't know if this guy knew what was going on, but anyway, now he's famous in Greenville, South Carolina, right? Famous. I found a bunch of pictures of rugby players in Eastern Europe, and I don't know, maybe that's something that rugby players need, you know, sloping foreheads and big brow ridges. But anyway, so these are obviously present in modern humans, and you could probably go down to the local mall and sit there for a few hours and see a bunch of Neanderthals for yourself. In fact, I was in a Bible study once with a guy who had a sloping forehead, large brow ridge, and he was pretty smart, you know, he was reading the scriptures and he knew what was going on. So anyway, here's a guy named Nikolai Valuev, very famous heavyweight boxer. He only lost a single match, very successful. And then he went on to be a member of the Russian parliament. He's a very famous guy, very intelligent man. And he actually really likes and enjoys people calling him a Neanderthal, of all things. So anyways, you can see the sloping forehead, large brow ridge. And so these are traits that are still with us today. They're not archaic. And so there's an example. So the guy in the middle is some kung fu guy. I don't know who he is, but you can see there's a sagittal crest down the center. And evolutionists used to say, oh, if a sagittal crest is on a Neanderthal or a Homo erectus skull, that would be an archaic trait. Well, it turns out not really. The guy on the left there in the suit is a world-famous neurosurgeon, brain surgeon. In fact, people go from all over the world to see this guy if they've got a really hard case. And he's a pretty smart guy, obviously. He's working on people's brains. And of course, the guy on the right, you know, he pilots a star cruiser across the galaxies. So, I mean, he must be pretty smart. So anyways, these traits are still with us today. And so I'm a tennis player, so I had to put this in there. So anyways, these two guys actually just retired. The tall guy's actually from Dallas, Texas. But anyways, this particular match went to several tie breaks. It was really close. But that one guy is over 1.5 feet taller than the other guy. And what is this? It's human variation, not evolution, right? It's just variation. And there's a lot of variation in humans. And so we're wrapping it up here. So Genesis 1:27 says, so God created man in his own image, in the image of God, he created him, male and female, he created them. And so why is that even important? Because we have this whole issue of a historical Adam being debated. So theistic evolutionists, in other words, Christians, people that claim to be Christians, at this organization called BioLogos claim that the earth is billions of years old, and that if there was an Adam, that he was selected as an archetype from a population of hominids, millions of, whatever. It's just ridiculous. Some of them don't even believe in a literal Adam. Other ones believe God selected some evolving human as an archetype. And so that really messes basically with the gospel. Yeah, you can still be saved and believe in the Lord Jesus Christ who died for your sins. And if you want to believe the earth's billions of years old and there was no historical Adam, but it is foundational to the gospel. We have to admit that it is key to the gospel message in the scriptures. And so as I mentioned before, we have these detailed genealogies and chronologies from Adam to Christ in multiple different lineages. One was done on Joseph's side, going all the way back to Adam. The other one on Mary's side, going back to Abraham. And so why is the Bible so specific about this? Why did two Gospels actually have these chronologies and genealogies in them if it wasn't important? Well, the Apostle Paul tells us in a number of places. So here's actually Romans 5. And this is just one scripture out of a whole section on this. Therefore, just as through one man sin entered the world, and death through sin, and thus death spread to all men because all sin. So how do you explain human depravity, right? Most of us would agree that humans have a sin nature. And yeah, we're not as evil as we possibly could be, but yet we still have this sin nature where we get angry, we get frustrated, we say stuff we shouldn't when we're in Greenville traffic, right? Or whatever, you get mad at your friend or your wife or your kids, whatever, do stuff you shouldn't do. But even more than that, how do you explain evil and corruption in the world? How do you explain death, evil, and corruption in the world unless you go back to Genesis and what Adam and Eve did when they fell in the Garden of Eden and disobeyed God and brought death, sin, evil, and corruption into the world? Hard to do without a literal Genesis or believing in that. In 1 Corinthians 15:21, for as in Adam all die, even so in Christ shall all be made alive. So the Apostle Paul said Adam, he talked about Adam being the first man, and then he talked about Jesus Christ being the second man, or the second Adam. So Adam basically means man in Hebrew. And so Jesus Christ was the second Adam. He completely fulfilled the law. In his act of obedience, he fulfilled every aspect of the Mosaic law and lived a perfect life, and then voluntarily went to the cross on our behalf and died for our sins as the second Adam. And so this is all critical and important to the gospel message. I don't think you can avoid that. And so anyways, I'm wrapping this up here. So I have a book called Chimps and Humans, and I did not pick the title to this book. Someone else did, but I did write the book, promise you there. So I've got articles or chapters in there on the DNA similarity issue, the chromosome fusion, junk DNA, pseudogenes, everything you could possibly imagine that evolutionists have tried to apply to the human evolution paradigm. I've addressed it in this book and debunked it. I have another book on the design and complexity of the cell, where I debunk abiogenesis, or the origin of life from nothing, which is one of the worst problems for evolution. We thought all the other ones were bad. And then I also talk about the amazing complexity of the cell and how that is engineered there. And my most recent book actually is The Fossils Still Say No, Paleontology of the Global Flood. So I chose that title because I was trying to honor Duane Gish. So Duane Gish is famous for showing that when creatures appear in the fossil record, they appear suddenly and fully formed with no evolutionary ancestors. In other words, there are no undisputed transitional forms between creatures. They appear suddenly, fully formed, and then the concept of stasis. In other words, they stay the same. So we can see creatures even from the Cambrian, the lowest layers in the flood record, the rock record, like starfish, brittle stars, horseshoe crabs. They look exactly like their modern versions. There's no difference whatsoever. Stasis, they've stayed the same. So that's great, but I wanted to say, well, why do we see certain creatures in certain layers and not other layers? And so I used the model of the global flood that our geologist has created, who was a geologist for Chevron for 10 years. And so he also worked as a professor in the Midwest as well after that. And he has literally mapped out the entire global flood all over the world. And he started in North America, and then South America, and Africa, and Europe, and Middle East, and Asia, and now he's finishing up Antarctica. And so his flood model showed, as well as the scriptures do, that it was a progressive global flood. It started in the oceans. That's why you see in the first few mega sequences in the fossil record, ocean creatures or marine creatures. And then when the flood waters start slamming into land and ripping up stuff and burying stuff, you start seeing tropical vegetation that would be living near the coast. You see all kinds of tropical plants. You see dragonflies. You see lizards, and amphibians, and millipedes, and things like this. And then as the floodwaters get higher, then you begin seeing creatures living at higher elevations. It's just basic. It's called burial by ecological zonation. And what's really interesting is that Henry Morris II, in his book, The Genesis Flood, which was written in 1961, put forth all of these ideas. How long ago was that? I mean, how many years? I think it was a long time ago, over 50 years ago. And he also said that the rock record, or the flood record, went to the very top of the geological column. And what's really amazing is that everything he said in that book in 1961 has now been fully vindicated by Dr. Tim Clary and his research that he has done. And so he has taken data from oil wells all over the world, exploratory oil wells, seismic data, rock outcrop data, every type of data that a geologist needs to go out there and look for oil that works in the petroleum, oil and gas industry. And he has applied that to mapping out the global flood. And over 70% of the Earth is covered by sedimentary rock or flood-based rock. And so creation science, in a lot of ways, yeah, the creation week's very important, but a lot of the arguments that you need to have when you're out there witnessing, sharing your faith, maybe you encounter evolutionists, hinge around the global flood. And so the global flood is key. And so anyways, actually Dr. Clary's entire flood model is actually in this book, as well as all of paleontology. And I also have chapters in there on soft tissue and biomolecules they're finding in fossils, including in the Cambrian. Not only dinosaur fossils have soft tissue, blood vessels with blood cells in them, including DNA that's been lighting up in studies. But we see this throughout the entire rock record because all of these fossils were buried and laid down 4,300 years ago in the global flood. And so in my book, I talk about that as well. I also talk about marine mixing because the floodwaters started in the oceans and then moved on to land. That's why you see sea creatures. That's why you see brachiopods, which is the shellfish. You can actually go to PaleoDB and see this for yourself. They're in every single layer of the geological column. And, excuse me, that's why you see sharks buried with dinosaurs. That's why in some of these higher elevation type deposits, you see monkeys, squirrels, bats buried with herring and skates. And so anyways, a lot of it's just common sense. But anyways, all that's in that book, which is, I think, going to be a key book for defending the faith among creation scientists for the years to come. And then we have another book, Creation Basics and Beyond, which basically has kind of everything in one book. The theology, the geology, the biology, paleontology. They're kind of in one book for good reference there. And that's only, I think, what, about $10 or something like that. So anyways, we have those resources, and I believe those books are still available up just one level as you walked in there. And we also have a Discovery Center in Dallas, Texas, which is super fun place. It's very high tech. We have a really nice planetarium there, as well as interactive displays and all kinds of things. And there's stuff there for adults. There's even stuff there for scientists. You can actually look into one of our labs where we're doing blind cavefish research, showing how creatures rapidly adapt to environments based on the internal programming God put into them. And so we have that and all kinds of cool stuff. We have an Ice Age theater where you can learn about the Ice Age that occurred after the global flood, which was important for creating land bridges around the world for people to disperse, and animals as well after the Tower of Babel, with just all kinds of stuff. So anyways, it's really an educational and entertaining place if you ever get to Dallas. So, we also have a magazine you can sign up for. We have a sign-up sheet just up there too with the books. You can get this every 2 months. It's got articles in there that are semi-technical, some are more lay level on creation science. Or you can just sign up and get a PDF and just read it on your iPad or whatever. So anyways, we have that available too. So anyways, that is it. Sorry if I went a little long here, but hopefully we'll have time for some questions. Okay. All right. So any questions? You all want to go home, I know. I've had enough.
Who won the tennis match, the tall guy or the little guy?
It's all barely. Yeah, he won it in several tie breaks. So, yeah. We should have invited him. He's just in North Dallas. Well, not. What did I say he was in this area for? I'm thinking I'm in Dallas. I'm not in Dallas.
Could you tell us some other example of things in the creation that would be 84% similar? Like a carrot and a hamster or something? I mean, what does that mean in some other part of the creation?
Well, yeah, I get that question all the time. So how similar are humans to rabbits, to squirrels, to mice or whatever? The problem is that it's very difficult to compare the human genome to creatures like that because the chromosome numbers are different. The gene neighborhoods on the chromosomes are different. And so, well, even between humans and chimps, papers have been published showing the gene neighborhoods, human compared to chimp, are totally different. Yeah, you can line up stuff if you pull it out of its place and then line it up. But, so that's the problem with comparing humans to other things. So I don't know if any of you know Matthew Cesardi, he's a bioinformatics guy. So him and I were actually talking about this because we get these questions in settings like this all the time. And so we tried to create a coefficient of similarity because of these problems with chromosome numbers and landscapes being different. And we tried to create this statistical coefficient of similarity, but it just didn't work. So we can't really say. Yeah.
Let's give him a big round of applause.